Dr. Daniel Amen is a psychiatrist, brain imaging expert, best-selling author, and medical director of the Amen Clinics. He has pioneered the use of brain imaging in clinical psychiatric practice and is a noted expert in the field of the brain and behavior. Dr. Amen's breakthrough research and insights provide hope and more effective treatment for millions of people today.
There are many people who are anxious or depressed or obsessive, prone to anger, they have attentional problems, and they thought their problems were all in their head, purely psychological, if you will. In the research that I've done, what we found is that these and many other problems are brain-based problems, and that when we optimize the brain, their lives become better. We often say, change your brain, change your life, and that's what we've seen. Today what we're going to talk about is something that's actually very cool. We're going to talk about brain development. We're going to talk about how your brain develops as you age. And I'm going to go over some brain basics. Now you've heard these before, but they really help us to introduce ourselves to the brain. And obviously the first thing, why is it important to know about brain development, is your brain's involved in everything you do. How you think, how you feel, how you act, how well you get along with other people has to do with the moment-by-moment functioning of your brain. So your brain development's really important in the kind of therapist that you want to become. Your brain has to develop properly in the kind of child that you are. And we're going to see children's brains are developing at an amazing pace, which is why you shouldn't stress them too much when they're little. But why you should also give them great opportunities for learning, because as we'll see, at the age of 5, your brain starts to prune itself. So the tracks that you're not using, the brain starts to lose. And why is it that we're starting to educate children at 5 years old? See, that may be a little late if you understand brain development. So who you are as a parent. Your parenting skills have a lot to do with how your brain has developed. And I'm going to teach you about a concept today called biopsychosocial evaluation. It's critical for you to know that. And when you think about treatment, biopsychosocial treatment. It's very important. So we're going to look at development from what we call a biopsychosocial perspective. So we're going to look at the biology, we'll look at the psychology, how you think, and we're going to look at the social situation that people are in. If you can understand those 3 circles for your patients, you do a much better job of evaluating and treating them. Okay? So first principle, your brain's involved in everything you do. The second one is when your brain works right, you work right. And when your brain is troubled, so are you. So again, these are SPECT scans looking at blood flow and activity patterns on the brain. The left is a healthy one. The right is someone who used heroin. It's why you don't use heroin. Just this week, we saw a 22-year-old who, when she was 18, had a heart attack. Can you imagine an 18-year-old having a heart attack? And she was unconscious and actually stopped breathing for about 10 minutes. And then they brought her back. So what kind of condition would that give you in the brain if you stop breathing for 10 minutes? Severe lack of oxygen. It's called anoxia. And if you look at her brain, it looks worse than the heroin brain. So oxygen is very important. So if you think of anything that could deplete or decrease the amount of oxygen you have, it's clearly toxic for the brain. And one of the things that we know decreases oxygen delivery to the brain is smoking. When you smoke, you constrict blood flow to the brain. What smoking does is it constricts tiny, tiny blood vessels. Now, why is that a bad idea? Because your brain's not going to get enough healthy oxygen, and it will prematurely age. Just like if you know someone who's been smoking for 20 or 30 years, well, they have skin that looks older than they are. Why? Because the small blood vessels are constricted to the skin. Well, the same thing is happening in the brain. Well, what about mothers who smoke during pregnancy? So this amazing organ that is at some points making 250,000 new neurons a minute, and you're smoking. And so as it's going through this incredible transformation, you're decreasing blood flow. What we know is that mothers who smoke during pregnancy, their children go to jail more often. Their children have learning disabilities more often, and their children have conduct and ADD problems more often. Okay? When your brain works right, you work right. When your brain doesn't, there's trouble. As we'll see today, clearly the brain is the most complex organ in the universe. It's estimated we have 100 billion neurons. Now, we actually have more at some time in our life. Now, neuron, same word for nerve cell, same word for brain cell, is the main active working cell in the brain. But it is surrounded by supportive cells. They're called glial cells. And the glial cells they help with nutrition, they help with vacuuming up toxic substances in the brain, so they are critical as well. Each neuron is connected to other neurons by thousands of individual connections. Some research says up to 40,000 individual connections between neurons, 40,000 individual connections within neurons. So more connections in your brain than there are stars in the universe. It's very important to understand that complexity, because as we look to see problems, you can just see why there might be problems in such a complex organism. 2% of your body's weight, your brain's the major energy user of the body. It's why what you eat matters. The food that you put into your body matters. Now, we're very worried in this country about a couple of things that have more than doubled in the last 10, 15, 20 years. ADD, autism, obesity, and kids wearing glasses. What is that about? And I think it has a lot to do with diet. And if you never think about your diet, think about that's what's going to feed your brain. So what should you stop kids from eating? Sugar. Because high levels of sugar are toxic to brain function. Some people think that President Reagan's little addiction to Jelly Bellies contributed to his Alzheimer's disease. But what do kids eat in the morning? Sugar cereals, cinnamon rolls, Pop-Tarts. I mean, think about, because our families are so busy and so much on the run, we feed kids what's easy. And even the things we think are healthy, orange juice, are really liquid sugar. So we have this developing brain that we're throwing all of this toxic stuff into. And do you think caffeine is good for the developing brain? What does caffeine do? Well, there are a number of brain scans in the literature. Actually, if you go to our website, brainplace.com, you can read about caffeine and nicotine, and what happens on brain scans is it dramatically constricts blood flow to the brain. So what do we do when kids come home from school? 4 Oreo cookies. So that's sugar and bad fat, because Oreo cookies have something in them called trans fatty acids. Sugar and bad fat, and then we give them a can of Coke, which is liquid sugar plus caffeine. So if the brain is really the major energy user of your body, and your brain is what really helps you to be you, why are we not more thoughtful about a child's diet? There's a commercial on television now for KFC. And it's like these two mothers in their minivan going, we're going to do something healthy for our children. And in the back, they're having this, fried chicken with applesauce, which is loaded with sugar. I mean, natural applesauce is certainly better than the kind you get at KFC, but it's loaded with sugar with the fruit roll-up, which is 90% sugar, and juice. Sugar. Sugar, sugar. It's bad. I mean, there's another way to do this. Again, brain is very soft. Skull is really hard. Mild traumatic brain injuries make a huge difference in people's lives. And interestingly, one of the most vulnerable times of the pregnancy is from the 5th month to the 9th month. So after your brain has really formed, in a sense, that those neurons are very sensitive to trauma. So you are never in your whole life at more risk than the day you're born. 3% of children are born with birth problems. And many of those birth problems happen the day they're born, whether they're breech or they're forceps deliveries or they have a lack of oxygen. And isn't it interesting, when you go into a delivery room, what the doctor does is he takes this little monitor and puts it on the baby's scalp. And you know what he's doing? He's measuring the baby's pulse, baby's heart rate. And the heart rate in a baby, and in you if you're healthy, is variable. It's always changing. If that heart rate flattens out, the doc goes, got to get that baby out because the baby's in trouble. We know that heart rate variability is a sign of heart health, whether you're a baby or whether you're an adult. It's actually something that we can train in biofeedback to have your heart rate be more variable. Because when you relax, your heart rate goes down, but it bounces around. When you're stressed, your heart rate goes up and flattens out. It stays at the same level. So the brain, unfortunately, is easily damaged. This is what a typical neuron looks like. We have to do a typical neuron. So we're going to talk about it. There are a number of different kinds of neurons. This is one called a pyramidal cell. Anybody have an idea why that might be? Because it looks like a pyramid. Okay. And it has things like dendrites. And dendrites are the bushy, tree-like things that are the antenna for the cell. It has a cell body. And in the cell body, there are many, many important things, like the nucleus that houses the genetic material of the cell, DNA, the code. This is the code that makes you who you are. And it has something called an axon. And the axon is sort of the output information for the cell. So if we look at it, dendrites, or the nerve cell's antenna, they're the receiver. The cell body has the nucleus, protein-making machinery. And what's very important and talked a lot about now in research is the mitochondria. Because the mitochondria are what produces energy for the cell. And many diseases, and many people think things like Parkinson's disease is one of them, is a disease of the mitochondria, where they become sluggish and can't keep up with the workload for the cell. And taking things like coenzyme Q10, which get the mitochondria perked up, help them work better, seem to be very helpful. So dendrites, receivers, cell body, making a lot of important substances for the cell, and the axons. Some of the axons can be long, like even a foot long. They're what sends signals out to other cells. There's something called an action potential. If there's enough input into the cell, at the junction between the cell body and the axon, there'll be an action potential, just sort of like a little thunderbolt, that then will race down the axon, and at the end of the axon will release neurotransmitters. So they will communicate, that cell will communicate with other cells. Something we'll talk about in a little bit. Axons are wrapped with a very important compound. It's made up of fat and protein, and it's called myelin. And myelinization, or the laying down of myelin is very important in development. So we will talk about that. Myelin, think of it as a protective sheath. So sort of like what's outside, you know, you put a sword in, a sheath. It protects the neuron. But there are little pinched out areas in the myelin that allow the electrical current from the cell body to the end of the axon to just travel faster. So they're called little nodes. Now, many of you have heard about gray matter and white matter. Gray matter, just a collection of cell bodies. White matter, why it's white? Because they're cell tracts that are wrapped with myelin. And that's what gives it the white color. There are things called interneurons. Those are neurons that connected close together. And projection neurons, those are neurons that are connected from far apart. So if you have local communication, that's with one set of neurons. So you think of the left hemisphere. Things are close together. It's the detail-oriented side of the brain. Lots of cell bodies. Things are close together. Versus the right hemisphere, where it's communicating with different parts of the hemisphere. That's why women often operate on hunches, because they're getting a couple of different areas of the brain working at the same time. So if we look at a cross-section of the brain, you can almost tell the difference between gray and white. And gray are where there's a larger collection of cell bodies, and the white areas are the myelin areas, the cell tracts, and the biggest one is the corpus callosum, which is bringing tracts from the left side to the right side. Again, larger in women, which is why women have greater access to both sides of their brain. If you take away all the gray matter and you just look at white matter, what you'll see, what looks like multiple tiny ropes in the brain. So you can imagine, if you have a very small stroke, stroke is where a blood vessel breaks, or blood clot goes and chokes off information. If you have a very small stroke in a certain area of the brain, nothing might happen. But say, for example, you stroke out one of those ropes, it might cause a severe problem, even though the stroke is no bigger than the end of a pencil. So it's where the stroke happens that matters, not necessarily how large the stroke is. So as they say in real estate, right? Location, location, location. You know, the brain communicates through synapses. As an axon ends, it often comes up against dendrites. Well, in order for the end of the axon to communicate with the dendrites, there are some electrical connections where, like, little thunderbolts will go across and it'll stimulate it. But most of the time, it's electrochemical. So at the end of an axon, as you see here, the axon terminal, the end of the axon, once the action potential gets to the end, it releases neurotransmitters. And the neurotransmitters go and lock into receptor sites. So you think of keys in a lock on the postsynaptic membrane, which just means presynaptic is the axon, the synaptic space. Postsynaptic is the dendrites receiving information. So the postsynaptic receptor sites are those receptor sites on dendrites. And if you get enough stimulation, then the dendrite will start to wiggle, and an axon potential on the other side of the cell body will fire off that axon. So that's how you get regular communication. And there are many kinds of neurotransmitters. We'll just talk about a couple of them. Acetylcholine is a very important neurotransmitter because what we find, it's involved in memory. It's excitatory. Now, some neurotransmitters are excitatory. What that means is it turns things on. Other neurotransmitters are inhibitory, turns things off, shuts things down. And some neurotransmitters like dopamine are both. In some places they turn things on, in other places they turn things off. Well, acetylcholine is mostly excitatory. And when there are low levels of acetylcholine, you can have things like myasthenia gravis, which is a muscle problem. Have Alzheimer's disease. In fact, one of the medicines we use to treat Alzheimer's disease are called acetylcholine esterase, which means an acetylcholine enzyme inhibitor, which means we inhibit the enzyme that breaks down acetylcholine. Because if you have too little acetylcholine in the brain and you want to get more, well, prevent its breakdown. And so the little bit that you have will stay around longer. And those medicines are very helpful in the early stages of Alzheimer's disease. But we've also found that they're helpful for kids who have learning disabilities, who have significant memory problems. Even in adults. I have one woman I gave Aricept to. That's one of these kinds of medication. And she said, Dr. Amen, about a month after she was on it, she said, you know, it was like going from 8 megabytes of RAM in my head to 128. How exciting. So acetylcholine, think of acetylcholine and memory. Serotonin, everybody's heard about. You know, Neil Diamond writes songs about serotonin. It's mostly excitatory. It helps to turn things on. And when it's low, it's usually a problem in a big way. People have problems with depression. They have problems with obsessiveness. They have problems with eating, with sleep, with pain. And what are the SSRIs? Selective serotonin reuptake inhibitors, which means once a cell releases serotonin, it prevents its reuptake, so it leaves more of it around. Okay? Well, what about dopamine? Dopamine is the motivation and concentration neurotransmitter in the brain. Now, in some parts of the brain, like the prefrontal cortex, it's excitatory. It turns you on. It helps you think. It helps you focus. It helps you concentrate. In other parts of the brain, it settles things down. We find it's low in Parkinson's disease. Anybody know somebody who has Parkinson's disease? It's where they shuffle. They have sort of a shuffling gait. They have a tremor in their hands at rest. It looks like they're sort of rolling a pill. Frequently get depressed. And after 3 years or so, there's a very high incidence of dementia. It's low in ADD. In fact, the medicines that we use, like Ritalin and Adderall, increase dopamine availability, and so people can concentrate better, and they have more motivation. It's too high in certain areas of the brain in schizophrenia. And so many of the meds we use in schizophrenia are called dopamine blockers, because they block the availability of dopamine. The new novel antipsychotics, actually, the newest one is called Abilify. It lowers dopamine in the limbic parts of the brain, the parts of the brain that sort of give you delusions and make you agitated, but it raises it in the dorsal lateral, outside, top of the prefrontal cortex that's involved in concentration. So before, things like Thorazine would make you less crazy but sleepy, and less sort of with it, would dull your personality, where the new ones, they make you less crazy, but you can still focus. You're not spending your day sort of zoned out. Norepinephrine is an excitatory neurotransmitter. When it's low, it's involved in depression. It's involved in ADD. It's involved in anxiety disorders when it's high. We block norepinephrine with certain blood pressure medicines. Interesting. If you're a cellist, but you're nervous, we might give you something like Inderal, which will block its function, and you won't shake when you are playing in a concert. There's a new medicine out. It's called Strattera. It's by Eli Lilly for ADD. And it's a selective norepinephrine reuptake inhibitor. Raises norepinephrine in the brain. And they found that it's helpful for ADD. The old medicine Dazipramine selectively works on the neurotransmitter norepinephrine. It's a wonderful antidepressant. So when we're targeting treatment for people, we're looking at brain systems, but we're also looking at what's the underlying neurotransmitter for that system. Epinephrine, many people know about it. It's the fight or flight neurotransmitter. If you see a rabid dog coming at you, your body produces a ton of epinephrine. So your eyes get big, or your pupils dilate, so the blacks of your eyes get big, so you can really see what's going on. All the muscles get shunted from your hands and your feet. They go to your legs and to your arms. You can either bite or you can get the heck out of there. So it's part of the fight or flight response. Epinephrine is part of the flight, part of the excited part of that response. And then two neurotransmitters most people don't really think about much, but we will be thinking a lot about them in the future, is GABA. Because GABA is the major inhibitory neurotransmitter in the brain. GABA is what settles things down. There's some new research just 2 weeks ago from Johns Hopkins that there is a subtype of ADD that is due to a deficiency in GABA. Well, we've known that for a long time. That's why for a certain subtype of ADD, we call it the ring of fire, where the whole brain is hot. Well, we give them anticonvulsants, anti-seizure medicines, because it raises GABA, settles things down. We also, there's a neurotransmitter called glutamate. Will be the year of glutamate because there's a new medicine called Namenda for Alzheimer's disease, which is a glutamate modulator, which means if glutamate is low, it raises it. If glutamate is high, it lowers it. And glutamate is the major neurotransmitter in the brain. It makes up 75% of neurotransmission in your brain. So of all the neurotransmitters that we just talked about, when you add them up, glutamate is 3/4 of them. So it's the major excitatory neurotransmitter in the brain. It's involved in memory. It's involved in alertness. But if you have too much, like you're under too much stress, or you're taking too much aspartame, or you're doing speed, or too much caffeine, all of those things increase stimulation to the brain. It's toxic for brain cells. And what Namenda does is it balances it. But mostly glutamate levels are low in people who have Alzheimer's disease. It's raising it. And some of the before and after scans that we've done on Namenda, very cool, because it seems to help balance brain function. The next brain basic is neurogenesis is alive and well. It used to be, when I was in biology, they taught us that you're born with all the brain cells you'll ever have. Wrong, not true. Your brain continues to make new neurons throughout your life. Now, many of your brain cells die, but it's replaced with new ones. The problem gets to be if you start killing more than you're making. Now, the problem can also be if you make more than you kill. What do we call that? A tumor. You're right. It's what cancer is. You're making more than you kill. So it's always a balance that we seek. So programmed cell death, apoptosis, is part of normal living. You just don't want to encourage too much of it to happen. Right? Now there's a term that's very important for you to understand. It's called long-term potentiation. What that means is when you learn something new, your brain makes a new connection. Well, the stronger you learn that, the stronger those connections become, so that when you know it, you really know it. So if you want to be good at something, you have to practice it in a good way. Because your brain doesn't care what it learns. It learns what it learns. So for example, if you're learning to play the piano and you practice badly, your brain will become very good at playing badly. That's why whenever you practice something, whether it's a psychotherapy and you're being videotaped, or it's learning how to play the piano, or it's learning how to sing, you have to practice it right. That's why coaches are so important. And you can't just do it a little bit. You have to do it until your cells get it right. That's why I'm opposed to very short-term kinds of psychotherapy. Short-term psychotherapy was an invention of the insurance companies. Because let's just think about it for a little bit. If you grow up in a traumatic family and you learn over time how to interact with people in a pathological way, do you think Successions is going to get long-term potentiation on a new way to do this? No. See? You have to be with somebody until you cause long-term potentiation for this new way of learning. It's also a critical concept at work, which is why you have to supervise somebody well enough until you know that they really got it. Because if you don't supervise them, they might not really learn it. And say they learn it for the moment, you learn it for the test, and then you don't have it anymore. It's gone. Right? I mean, isn't that sort of frustrating? You crammed for the test, and 6 years later, it's like Krebs cycle. I heard about that somewhere. So long-term potentiation, very important. What you learn. I mean, practice in a good way and to learn what you want. Learning and physical exercise, they encourage neurogenesis. Whenever you learn something new, your brain makes a new connection. We'll talk about this later with Hebb's rats. It's a funny story. Some of you read the textbook, the chapter this week. Hopefully you read it. And looking at development, the brain is actually relatively low in activity at work. From 0 to 3, it just ramps up dramatically, all the way up till 5. And then at 5, it starts to killing itself off. It prunes itself. Now, pruning is very important. I mean, why do we prune rose bushes? Does anybody know? Why do you prune rose bushes? I'm sorry? So the healthy part grows. You don't want all the branches that really aren't producing very much. Well, I think the brain has the same concept, but it's sort of a use it or lose it concept. Now, your brain continues to be very active until you're 40. And after 40, the repair mechanisms in the brain become significantly less, which means we're sort of programmed to live until our children are able to take care of themselves. And after that, you're sort of on your own, which is why really working to keep your brain healthy after 40 is critical. Okay. As I was looking at this lecture on brain development, miracle was written all over it. I mean, it's really a miracle. The brain is constructed in different levels that have different functions, which we'll talk about. Development is influenced by genes, no question. Your genes matter. But if you look at all the trillions and trillions and trillions of connections in the brain, there are not that many genes. So your genes sort of lay down the roadmap. Your experience has a lot to do with how those roads get filled in, and whether some of these roads are dead ends or whether they become super highways. So it's never, and never let anybody box you into the idea it's nature or nurture. It's both. It's always been both. But we used to think there were 100,000 genes. And when they finally finished the Human Genome Project, they went, there's only 30,000 of them. I mean, which I think is quite a large number. But it's really not nearly as much as they thought. And so they're thinking the blueprint is really less predetermined, if you will, than we thought. Experience in life makes a big difference. And just have to think about the Romanian orphans. We've actually scanned a number of Romanian orphans that were severely neglected. And they have severe problems. So their genes might not have been terrible. But if you neglect somebody for the first year of life, their brain is not going to develop in a healthy way. So development's influenced by genes, it's influenced by your environment, and by your experience, by who's singing to you, who's talking to you, who's training you. So hopefully, your parents have good frontal lobes, because your parents become your frontal lobes until yours fully develop. And at what age do frontal lobes become fully developed? 26. So we're letting kids out way too soon to fend for themselves. So as the brain increases in complexity, if you look through animal species, the brain can do more complex tasks. Different parts of the brain develop at different rates. When you're 2 months old, your occipital lobes in the back involved in vision really ramp up. So vision is not that great early on, but by the time the baby's 4 months old, vision's starting to get pretty good. The frontal lobes don't develop until much later. So children do not have the same social skills as adults. And this is an image, a picture from the book, from the Kolb textbook, is that social development is different. So when children are asked to say the emotion of the figure with the blank face, they're not nearly as good at it as adults are. Because empathy is something that is not really ramped up until the frontal lobes are fully evaluated. So if you're getting after your teenager for being selfish, it's normal. Their frontal lobes, that part of their brain involved with empathy, is not as well developed. Some stages of brain development to look at: cell birth, neurogenesis, and something called glial genesis, cell migration, cell differentiation, cell maturation. Your brain matures. Synaptogenesis. Just think of these are like big words. Just sort of break them apart. Synapses, making synapses in the brain. Cell death, pruning, and myelogenesis. That's the thing, laying down myelin over time. And as you lay down myelin, you increase the efficiency of the neurons working. So cell birth, on the day of fertilization, when a cell comes together, it's one cell. And then it becomes two. And then it becomes a disc. And by day 21, we see the neural tube start to form. That disc starts to fold in on itself. And on day 23, it closes. And actually, that's sort of where the beginning of the cerebral cortex starts to happen. So these are images, again, from the book. And what you'll see is the neural tube start to form. And as it forms, it puts inside this tube are what we call stem cells. Now, I don't know where everybody is on the whole stem cell question. It's pretty interesting. But think of it as the nursery for the brain, where initially these are undifferentiated nerve cells. And it's over time, through certain genetic programming and chemical interactions, like how many male hormone interactions you get, it'll determine where the cells go and how they will grow. So the cells lining of the neural tube is the brain's nursery. So stem cells, brain's capacity for self-renewal. Cortex begins development at 7 weeks, completed mostly by 20. So 5 months preemie can live outside of their mothers. You really can't live before 5 months. That's pretty amazing that 26-week-old babies preemies can live, where 20 years they couldn't. Between 5 to 9 months of gestation, it's a very vulnerable time of the brain. Anoxia, which just means lack of oxygen, toxins. And why they think it's worse at 5 to 9 months and not before is because a lot of the cells have already been laid down. So if you kill them, that's after that stage in development. Cell migration. Cells migrate. It's like the glial cells, the supportive cells, will lay down these roads, and the neurons will then follow them. And you've probably all heard that the brain sort of is, the cortex is in 6 layers. Well, layer 4 is the input layer. That's where the sensory information to the cortex comes in. Layers 1 and 3, think about it. They sort of integrate the sensory information. And layers 5 and 6, that's what tells other parts of the brain what to do. Cell differentiation, you have stem cells that then will either go into neural cells or into glial cells, and into different kinds of neural cells, like interneurons or projection neurons, or if different kinds of glial cells, supportive cells. Along with cell maturation, if you look at this image, what you see is the one to the left is a newborn. There's not much going on yet. And at 1 month, things are starting to fill in. In 3 months, you can see they're starting to make more connections. And the axons are really growing fast. And at 6 months, a lot more, at 15 months. And it sort of follows what babies can do. And by 2, they're already saying no, right? I mean, their brains are real. I'm not going to do it. I don't have to do what you say. You're not my boss. I mean, by 2 years old, that's in part because of their brain. So, with maturation, you'll hear words like dendrite arborization. So, do you think of arboretum? Hey, that's where branches are growing. That's where the dendrites are becoming bushier. It's a fabulous study from UCLA. Arthur Arnold Schliebel. He looked at teachers and migrant workers. And he looked at autopsied brains. And what he found with teachers is that arborization in the language centers, it was just flooded with dendrites, with these bushy endings to them. So they had lots and lots going on in the language. And in the migrant workers, it was like a desert. It sort of looked like Barstow in the language center. But if you look at the physical activity center of teachers, Victorville, where the workers had lots of activity. So it sort of is use it or lose it. If you're using language a lot, that part of your brain continues to be active to sprout new dendrites, have more cells and more cell contacts in those areas. Where if you're not using it at all, like, please don't ask me to fix your car, because I have nothing in that part of my brain. In the car fixing part of the brain, there's like nothing that's there, because I don't use it. You know, that's not something that I do. So dendrites continue after birth. They grow slowly, heavily influenced by experience. Axons, the part going out from the cell body, they grow fast, sometimes up to 1 millimeter a day during their initial growth spurts. There are many growth factors involved. In fact, they're using growth factors in the treatment of many neurological disorders. I have a colleague over at UC Irvine. Jim Fallon is using something called TGF-alpha. And TGF-alpha really helps neurons to grow. And so they're thinking about using it with Parkinson's disease and Alzheimer's disease and so on. And experience is heavily involved. So Hebb's kitchen rats. Hebb is a famous neuroscientist who he raised some rats in the lab and just gave them the stimulation that you would get in a cage in a lab. And a whole other group of rats he took home with him. And they lived in his kitchen. And his wife was not happy about this. Can you imagine? The rats are living in the kitchen with them. But the rats in the kitchen had a much more varied experience, especially with Mrs. Hebb and the broom. And the cats. So they had to be more creative with their food. They had to be better at problem solving. You with me? And the rats in the kitchen, when he would slice their brains, had significantly larger brains and brains that had more densely packed neurons. Okay? You do everything for your child, they will have a lighter weight brain. Do you get that concept? If you provide somebody with everything they need, their brain will not learn how to use it. So, just some images of what the brain looks like at different parts of development. And what you see is, it's a sort of this blob early on, and it forms more and more and looks like a brain. But what we see at 5 months and 6 months and 7 months, 8 months and 9 months, is really becoming much more detailed. And all of those ridges, and called gyri and sulci, it's developing more area so that you can sort of learn more information. And fetal alcohol syndrome smooths out the brain. So it takes away, that toxicity takes away the development and the ridging that is crucial to brain health. Synaptogenesis is just the brain making synapses. And if you take 10 and put 14 zeros behind it, that's about how many synapses that you have. Too many for genes alone to determine it. So it lays out the general guidelines, guided by clues and signals, hormones, experience. Simple connections at 5 months, complex at 7 months, in utero, increases dramatically after birth. Synaptic death and synaptic pruning. I really like this image from the book. And why would the brain prune itself? Well, if you think about the brain in a block of sand, well, it really needs to take away the things that it doesn't need so that it has the best shape and the best function. So there's an overproduction of neurons and synapses, and subsequently we lose about 42% of our brain. Now, if we don't lose enough, that's one of the theories for autism, is there's not enough synaptic pruning. If we don't lose enough, there's too much going on in their head and they can't think. They can't focus. Brain prunes unnecessary or incorrect synapses. One study said you may lose up to 100,000 per second. Experience, opportunity, stress have great influence on this. So there's normal programmed cell death. You just don't want it to go too far. And if you smoke pot, you encourage it. If you drink too much alcohol, you encourage it. If you're under chronic stress, you encourage it. Just looking at the chart and synaptic pruning, what you see is there's just dramatic, from birth to what? To a year old, just dramatic growth in the brain. And then it drops off a little bit, and at 5, another dramatic drop. A little bit more from the time that you're 10 to the time you're 20, it grows a little bit. And from 20, it's sort of a downhill course. But what's interesting in the normal study, in our normal study, the people who really take care of their brain have healthy brains as they age. The people who are not nice to their brain, even though they're, quote, normal, their brains look ugly. So care of your brain, one of the things I hope you get out of our time together, caring for your brain is critical to keeping it healthy as you age. Myelinization. Myelin wraps and insulates the neuron. It increases efficiency of how they function. Begins after birth, continues all the way into adulthood. Maturity is associated with how much myelin is laid down, because that's how your brain's really working more efficiently. The last place to develop is your brain's supervisor, the part of you that watches you, the part that helps you with conscience and forethought. Drug abuse damages myelinization. So it damages maturity. So you've known people who've used drugs if they start when they're 15 and they stop when they're 25, emotionally they still may only be 25. It's really an important message to get to children and teenagers and young adults. This is what myelin looks like. If you look at these rings around this particular neuron, it's wrapped with these coats of substance myelin. So just to think of it almost like plastic sheeting on a copper wire. It's absolutely critical. And there's an illness called MS, multiple sclerosis. And what is multiple sclerosis? Where you get deposits of certain plaque-like materials that rip off the myelin. So it's a disease of myelin, if you will. And so then the neurons start to short circuit. They don't work properly. Okay. I told you we'd talk about this. It's a critical concept. It's one of these like top 5 things I want you to leave this course with. It's a biopsychosocial influence on the brain and a biopsychosocial to approach. What do I mean by that? When you evaluate somebody, you have to look at their biology. So in many ways, that's what we do in this class. We're going to look at the biology of the brain. You also have to look at their psychology, how they think, what their development was like. And it's key to understand the social situation they're in. So when you look at the biological influences, what might those be? Well, genetics. Genes are very important. Mental illness runs in families. The best way to build an adult mental health practice, see children. Because then you see their parents. And you see their grandparents. We know ADD runs in families. No, depression runs in families. Alcoholism runs in families. Criminal behavior runs in families. Your mother says, marry somebody from a good home. She's right. Although now that I'm almost 50, I look at her and I go, so where are they? 49% of the U.S. population at some point in their life has a diagnosable psychiatric illness. Right? As we're doing this study on, quote, normal. You're doing the study on normal. There are just not that many people. We have screened maybe 700. And today I have scanned 37. We need more. Has anybody turned in their paperwork? We need more. All right, genetics. Prenatal exposure. So what kinds of things might that be? Prenatal exposure. To cigarettes, to alcohol, to cocaine, to redoing the nursery, the baby's nursery. They paint the nursery right before the baby comes home. This is a very bad thing. Mother's in there painting the nursery. Or they put new carpet in the house, which is filled with toxins. Now, many mothers go, oh, I didn't drink when I was pregnant. As soon as I found out I was pregnant, I stopped. So when do women find out that they're pregnant? Anywhere from 2 weeks to 2 months. And when is the most vulnerable time of the pregnancy? It's the first 12 weeks. I mean, we talked about the last 4 months is really a vulnerable time. But when that neural tube is forming, which we said is like 3 weeks, you know, it's a little dicey. Right? So many people who are really in trouble, no one has the history that they had prenatal toxic exposure. It's a little unfair. How your health is, your physical health, whether you have thyroid illness, whether you have anemia. I did one scan once on a woman her brain looked like she was a heroin addict. The problem was she never used heroin. And I knew her very well. She was my best friend's wife. And I'm like, something's toxic here. And so I ordered lab workup on her, and she had pernicious anemia, which is from a B12 deficiency. So she had severe anemia. that was causing the toxic look in her brain. Liver problems, kidney problems, pancreatic problems, heart problems. I mean, all these kind of medical problems can cause significant brain problems. Diabetes, we know, is very bad for brain function. Toxic exposure. Can be from substance abuse, can be from living on a farm, can be from being around secondhand smoke. Can be from lots of things. I had one patient who drank a bottle of lye. Very bad for your brain. Even though it ripped out his esophagus, they had to take part of his intestines and transplant it to make it his esophagus. It's very bad. Trauma. Physical trauma. And emotional trauma. Brain system health, how healthy your brain systems are, and how much neurotransmitters that you have available to you. Okay. I want you to be thinking psychological also means biological, because if you are bonded properly, your brain is making these connections. So it becomes a physical, not just emotional experience. You need proper stimulation. Not enough stimulation, your brain won't connect itself properly. You have too much stimulation and you're exposed to chronic levels of stress hormones, your brain will start disconnecting itself, or it will connect itself in a random way. How you talk to yourself matters. If you talk to yourself in a negative, harsh, evil, awful way, I call them ants, automatic negative thoughts, the thoughts that come into your mind automatically and ruin your day. If you have a high ant population, your emotional center will be flamed. And where is it that we teach children how to talk back to the bad thoughts in their head? We don't. We teach them factoring and algebra. When have you ever factored as an adult? I've never factored as an adult. I helped my 16-year-old factor with her algebra homework, and I'm thinking, maybe we could teach them more practical things. How you talk to yourself, the experiences you have, and your brain develops in large part on how effective your mother's brain is, and how effective your father's brain is, and how effective your teacher's brains are. So it's brains interacting with brains. But if you're in an environment with ineffective brains, your brain's not going to be humming as good as it could be. Right? So your support system matters. Your level of stress, moment by moment. Now, we all need a little stress to sort of keep us moving. You know, coming up, there's a paper due. Right? So you know it's due. So you're like thinking about it. It's going to push you along. You get 12 papers at once. It's too much stress. You can't handle it anymore. It's very bad. So support system, stress, your opportunities. I mean, if you're in a really great program that gives you lots of opportunities for learning, your brain will be better than if you like the only thing you're exposed to or, you know, chronic alcoholics at the VA. That's it. It's the only thing you ever saw. You're probably not going to be that effective in treating, you know, patients in Newport Beach. The models that you have, the models that you have as teachers, the models that you have as parents, the models that you have as friends. That's why you become like the people you spend time with. So make sure you're spending time with people you want to be like, rather than, you know, I always tell the teenagers I see, if you're hanging out with drug dealers, you're going to be a drug dealer. Right? If you're hanging out with people who are doing well in school, odds are you're going to pick up that same mindset. People are contagious. So when you think about the key to diagnosis and treatment of patients, don't just think in the DSM-4. The DSM-4 is an artificial document. The DSM-4, people got around in committees and they voted on stuff. You know, this is an illness, this isn't an illness. You know, PMS, well, maybe next time, but not this time. You ever been around women that had severe PMS, you better believe it's an illness. I've seen scans before and after, it scares me. But think about it in a biopsychosocial way. What are the biological causes? What are the genetics of this client's problem? What environmental toxins might be? What's their health like? What's the drug abuse situation or the caffeine or the dietary situation? All of that are biological influences. Psychological influences, what do they grow up in? How do they talk to themselves? What is their long-term potentiation for how they talk to themselves and how they interact with others? Right? I mean, if you've been talking to yourself really badly for 45 years, do you really think 8 sessions of cognitive behavioral therapy is going to change those neurons? No. Of course it'll help, but you need to then stay with them for a year or 2 years so that they get it in their head, so they got new neuronal connections in their head. And the social situation you're in, they're in a job that's killing them, talk to them about that. Or if they're fighting with their spouse, get their spouse in, or get them into the right kind of environment. If their kids are driving them crazy, get everybody evaluated. It's the rule in my head, everybody in the family. If I see one person in a family, odds are I'll end up seeing everybody, just because I want to know who's contributing to what part of the problem and who can be part of the solution. Right? Because often in families you have the really terrible child and the perfect child. And they're both working on each other. And if you understand it, things can be so much better. The question is about, can forceps cause problems? Well, putting the little monitor on their scalp I don't think causes a problem. It's just monitoring it. Forceps only cause problems, in my experience, when they bruise the baby's skull. If you get bruising, odds are underneath where they've slammed the brain, squeezed the brain together, they may have damaged the frontal lobes or may have damaged the temporal lobes. And now they're using a vacuum more than they use forceps. And you just have to sort of think, what's the pressure going on? It's one of the reasons that in many, many states, OBGYN doctors can't get insurance, because people are coming back and suing them 10 or 15 years later because the child has learning problems and they think that somehow what they did interfered with it.